So, welcome back. Neuroscientists are getting pretty good at relating the big five personality traits to the brain, in particular to the important role played by the gaps between neurons called synapses. So neuroscientists and geneticists have looked at the genetic variation that exists between individual people that affects how synapses are used or how effectively or ineffectively they might be used in certain systems in the brain. Neurons speak to each other across synapses using neurotransmitters. There are about 50 neurotransmitters and they allow our nerve cells to communicate with each other. But I'm going to focus on serotonin and dopamine as they're both involved in mental traits that affect our personality in very direct ways. So let's take neuroticism as an example, one of those 5 dimensions I mentioned. Neuroticism is a good example for us because it relates to politics, which I just talked about. Now, neuroscientists know which parts of the brain are involved in the response to threats. There's a circuit involving a structure in the limbic brain called the amygdala, which is shown here in red. The amygdala is involved in your emotional responses to the things you experience. So if someone invades your personal space and you feel an unease, that's the amygdala squeaking away inside your brain. The amygdala is connected via nerves to the cingulate cortex, which is shown here. These nerves form a circuit so that information can pass from the amygdala to the cingulate cortex, then from the cingulate cortex back to the amygdala. Your brain is full of circuits like this. Anyway, the cingulate cortex is one of 3 areas of the brain that are critical for self-awareness. And one of its jobs is to think about what the amygdala is doing. So if you go to a frightening movie, the amygdala feels threatened and it starts firing as if you're in danger. The cingulate cortex, working with your frontal cortex, is going to reply back, "Take it easy, it's just a movie." People who suffer from post-traumatic stress disorder, they've got a lot more amygdala activity than they should have and less cingulate cortex activity. Now, the synapses of the nerves transmitting messages between the amygdala and the cingulate cortex use serotonin to connect them. And a lot of the variation in how our amygdala and cingulate cortexes function relates to how the genes involved with serotonin are regulated. So remember that genes consist of more than just the sequence coding for a protein. There are also promoter regions in front of the coding part to which other proteins are going to bind and that account for how much of a protein is made, and when and where in the brain it's expressed. So because brain function is specified by circuits, even small differences in the location of brain cells or the amount of a particular receptor or enzyme can produce large differences in function. I'll give an example of how this might work. There are 2 common forms of the serotonin transporter gene, which codes for the protein that removes the serotonin from the synapse and returns it to the original cell. And they differ in their promoter regions, so that one form of the promoter is much better at getting transporter produced than is the other. So some people have more transporters and some people have fewer transporters. And the point here is that if you produce a lot of transporter, your signal across the synapse will be fast, quick, and then over. Any single gene's impact on mood or behavior is going to be limited, of course, and these single gene or candidate gene studies must be viewed with that in mind. Yet many studies on these genes suggest that people, monkeys and rodents with a short or low activity form of the transporter gene are more sensitive to the environment and tend to suffer significantly more anxiety and depression when faced with stressful life situations. Brain imaging has shown that when stressed, the cingulate cortex and amygdala light up differently, and the circuits that connect these 2 areas are weakened in those with the low activity allele. Now, if these studies are correct, the ultimate effect of the low activity alleles is that the part of the brain that's supposed to be dampening down your fear responses doesn't do the job very well. Well, you might ask yourself, why would the low activity allele have survived in human populations? Well, perhaps being low stress and happy is overrated if what you're trying to do is survive in this world. Researchers with that particular worldview say that having a high activity allele lets you see the world through rose-coloured spectacles because you filter out bad news. So depending on your attitude, which will itself be determined in part by your genes, you could say that the low activity allele isn't about making you prone to anxiety, it's about giving you a more realistic view of the world than the high activity allele. Okay, putting aside neuroses, you'll probably notice that your worldview is particularly slanted when you're in the intense throes of romantic love, and particularly during orgasm. Well, brain scans show that an orgasm lights up 30 parts of the brain with a massive amount of activity in the cingulate cortex, while it smothers other parts of the brain like the amygdala. Intense romantic love and orgasms release serotonin and feel-good chemicals like the so-called cuddle hormone, oxytocin, and its close relation, vasopressin. It's believed to be the oxytocin that smothers the amygdala, and that this increases generosity, trust, and empathy. Well, unfortunately, that's not an unalloyed good, because the amygdala is the threat detector. It starts firing when you see danger, risk, uncertainty. So the upshot of dampening the amygdala is that the brain in love is prone to bad decisions. It's got trouble detecting threats, like a jealous spouse, and connecting actions with long-term consequences, like the effect of unprotected sex, for example. Now, there's a maxim in biology that for every problem, there's going to be some animal on which it can be most conveniently studied. A control model system. Now, a lot of what we know about the impact of hormones on our love life was first worked out in some rodents called prairie voles. Now, prairie voles like these live in couples, and both father and mother care for the young for many weeks. Now, montane voles, on the other hand, which look pretty much identical to prairie voles, they're much more typical of mammals. The males are promiscuous and they take no care of the children. It turns out that the difference between these two closely related species lie in the promoters of their molecular receptors for vasopressin and oxytocin. The receptor's job is to fire up the neurons in response to the hormones. The pair-bonding prairie vole has far more receptors in various parts of the brain than the montane vole. In a clever series of experiments, researchers took the prairie vole's vasopressin receptor gene and using gene therapy inserted it into the brain of the philandering montane voles, which turned those little Casanovas into faithful husbands and fathers. Now, although montane voles and unfaithful husbands have some key differences, the general mechanisms may be similar. We've got several vasopressin alleles that differ from each other in their promoters, and at least one of these alleles is associated with poor pair bonding and promiscuous behaviour. You can now get genetically tested to see which variant you have. There's talk about developing drugs that can modify a man's vasopressin receptors. So think about that. It's a brave new world. Well, as we're on the subject of orgasms, twin studies by Tim Spector's team, and Tim Spector is a king of twin studies, have shown that the ability of women to have orgasms has a strong genetic component. There's an extreme condition called restless genital syndrome, where the nerves supplying the genitals are overexcitable, and a woman can have up to 100 orgasms a day. That's a common mechanism with the much commoner restless leg syndrome, which is about 60% heritable. Now, some of the genes responsible for both conditions affect the actions of nerves that use the neurotransmitter dopamine. Dopamine is involved in all kinds of rewards, things that make us feel good: eating, drinking, winning a football game, making money, buying a new car. It's the motivation chemical which fuels our cravings. Part of our brain that require dopamine are then not satisfied, and other parts of our brain, when carrying out functions essential to our lives and reproduction, they release dopamine to reward us. Oxytocin is also about reward, but it's usually more social. It's the reward of being around friends, of going, when you see a baby. It's about bonding and trust. Now, there are times when you have the combination of oxytocin and dopamine, like when you're making love. Now, just as with serotonin, there's a lot of variation among people in the functioning of the dopamine system. There's variation in how much dopamine is produced, and in the dopamine receptors on the synapses that pick up the signal and transmit it. There's 5 different dopamine receptors, and different parts of the brain use different dopamine receptors to receive the same dopamine molecule. So having so many receptors allows different systems in our brain that all use dopamine to evolve independently of each other. There's genetic variance in these receptor proteins that cause the receptors to bind dopamine less efficiently. That means there is less signal coming across the synapse. When these happen, the people with these mutations are stimulated to crave more stimulation, and this causes novelty-seeking behavior, which can lead to risk-taking. It's really dopamine craving that is leading us to seek novelty and risk. Now, if we consider whole populations, there may be a benefit in it of having individuals who are risk takers and people who avoid risks. For the individual, the golden mean applies. If you avoid all risk, then you're unlikely to achieve anything. If you take risks all the time, you're probably going to get yourself killed.